A little on the genetics of beans. It's a diploid, self-pollinating crop. It's an annual species. A diploid where the 2n is equal to 2x is equal to 22. And we have 11 chromosomes. The haploid number is 11. We have 11 chromosomes, and those are numbered here, PV for Phaseolus vulgaris, 1 through 11. There's very little natural outcrossing in beans, so the varieties we produce are not able to produce hybrid beans as such. We're producing pure line, near homozygous pure line varieties. In terms of the information on bean genome, it was recently sequenced in 2012. Beans have a relatively small genome size of 500, estimated at 587 megabases, and there's substantial genetic synteny with soybean. Even though soybean has 40 chromosomes, there was a duplication, but there's still a large amount of genetic synteny between beans, common bean, and soybean. Looking at the MSU dry bean breeding program, I've listed here the major research objectives in our program. This would apply to many programs, not only bean breeding, but other crop improvement programs. Our focus being on yield and processing quality. Those are essential criteria for us. And then we also work with disease resistance, architectural improvement for an agronomic adaptation, and increasingly we're looking at stress tolerance and drought tolerance. And then the last one I list there is grower friendliness, and the picture on the bottom of the screen illustrates two varieties, one on the right which has retained its leaves at harvest, one which has dropped its leaves. The one on the right would require the use of a desiccant, and again a grower-friendly variety would be more likely to be the one on the left-hand side of the photograph. Production in the state of Michigan is located in the central part of the state, up through what we call the thumb of Michigan, as you look at the back of your left hand, and that's the major production zone for dry beans in Michigan. In terms of the breeding systems that we work with, the main breeding system that we use in our program is the pedigree breeding system to focus on yield and quality traits, but we have used a series of other breeding methods as needed. Backcross breeding to introduce disease resistance, a number of years ago to improve architecture, as is illustrated in the photo on the right, to develop an upright type from a more decumbent prostrate type. We used the system of recurrent selection. We have used inbred backcross methods to introduce diversity from the distinct gene pools. And over the years, we have developed some markers for marker-assisted selection, largely for disease resistance, and I should say for qualitative disease resistance. Like most breeding programs, then, we have focused on more QTL analysis for quantitative traits in more recent years, and as we develop more marker technologies, particularly SNP markers, now there's a number of projects now starting to look at genome-wide association mapping to improve quantitative traits such as drought tolerance, insect resistance, certain disease resistances, and also improving biological nitrogen fixation. This is the bean breeding pyramid in which we kind of structure our program. We're expected to produce varieties for our growers here in Michigan or for beyond the borders of Michigan. So in order to develop varieties, new varieties will surface from the apex of that pyramid, which we largely use our pedigree, a system to develop those varieties. But to undergird that, we need a broad genetic foundation in the base of that pyramid. And that's where many of our graduate students would be working on projects in terms of genetic inheritance of specific traits, on QTL analysis, or initiating some of these studies on GWAS as a way of introducing new variability, understanding it, and integrating it into these different seed types, and then moving it up through the different systems of breeding through the intermediate level to the apex level, where we would be focused largely on yield improvement. Within the constraints that I list here, constraints of growth habit, maturity, we work with a shorter season crop and with certain disease resistances, and then the quality traits that are very critical. Market class traits for the sale of the seed within the industry, seed traits in terms of size, shape, color, and then canning quality. Since all beans have to be processed, canning quality is a very is an essential criteria or trait as part of our breeding program. So looking at the breeding timeline for variety development, when we're asked how long it takes to develop a variety, we indicate generally we're working on something like a 10-year program. For the first few years are involved in our initial crossing, early generation selection. As I indicate, we do a lot of single plant selection through the pedigree system in the early generations. And to hasten the program, we conduct a shuttle breeding program with winter nurseries in Puerto Rico. Then in the second trimester, years 4 through 6, we're dealing with yield testing and replicated trials, and everything that goes into yield trials is evaluated for canning quality. And then as materials would emerge and look like promising as new varieties, then we're still looking at 3 years of seed increases from breeder through foundation and certified before we could market a new variety in a 10-year time frame. If we're looking at the crossing of beans, or here we're the bud pollination of beans, the photograph on the right-hand side shows an open flower. When the flower is opened, it's already pollinated, which therefore requires us to do bud pollination. And you can see on the photograph on the left-hand side, we're opening that flower bud rather carefully to access then the petals. And this is a legume flower. Therefore, when you can extract the banner and wing petals, you can actually press down on the wing petals, as is illustrated in the upper slide, with forceps, and that trips the flower, and it forces the style and stigma out of the filament, away from the anthers, and it doesn't retract, it stays out. So you can conduct pollination without emasculation. Then once the stigmatic surface is exposed like that, you'll simply go to the open flower that you'd be using as a pollen source. You would trip it in the same fashion, and then you can remove the style stigmatic surface, which has pollen laden on it, and transfer the pollen simply as illustrated in the bottom portion of the slide by hooking that style from the pollen parent, which has the pollen coated on it, hooking it onto the stigmatic surface of your female parent. And with this kind of relatively non-invasive bud pollination, we can get over 90% success rate in pollination. Pollination is conducted in the greenhouse, as illustrated in this breeding timeline. We usually are conducting our crossing in the fall of year 1. Those crosses then are selfed during the spring. So each year we're going to the field with a new group of F2 materials, and that's where we initiate our pedigree selection, or our SPS, or single plant selection. We do a lot of single plant selection because we're looking at a range of plant agronomic traits, plant architectural traits, and seed traits. And then we move that material to Puerto Rico in the wintertime, where we conduct generally mass selection for more convenience on the F3 plant rows, and those then come back to Michigan the following summer as F4 plant rows, and there we continue with single plant selection at that stage. And that's usually where we end our single plant selection, at the F4 generation. We proceed then with the second season in Puerto Rico with the F5 generation, and that material then that's selected. So we're selecting in both Michigan and Puerto Rico as part of our shuttle breeding program. Those then enter in year 3 yield trials as F6 generation materials in our preliminary yield trials, and then materials get advanced. And as I indicated earlier, materials that are evaluated in yield trials then are also processed or canned, so we can evaluate for canning quality. And then towards the end of somewhere around 3 years of yield testing, we have some idea of materials that might have certain potential as new varieties, and those are identified usually as early as that stage. And here we're just illustrating that shuttle breeding. In Michigan, we're showing some of the preliminary yield trials in the upper part of the slide, a single plant selection in the F2 and the F4, and then in the bottom slide, in collaboration with the University of Puerto Rico, then we're conducting then our mass selection of plant rows in the F3 and the F5 generation. Materials then that would be selected at the end of testing in year 6, then we enter as pre-breeder materials, and since we have certain endemic diseases in the eastern U.S., including Michigan, we establish a breeder seed source in the western U.S., usually in the state of Idaho. Our university does not have farm there, but we contract that with local companies and growers. And then we're illustrating then 3 years of seed multiplication from breeder foundation through certified, with then a distribution of commercial seed in year 10 to our growers. I want to talk a little about some of the improvements in plant architecture that we have been involved in over the years. Here I'm illustrating the 4 plant types that you find in common bean. The determinant type on the left-hand side, which we call type 1, is a typical bush type. Many of you would be familiar with this as a garden bean type that you may be growing in your garden for horticultural, for pods and so forth. The one in the middle is the other 3 types are all indeterminate with different levels of vine extension. We have the type 2, which is an upright, more similar to a soybean type, and then we have the type 3 vine type, which is more adapted to dryland western production areas. And then the type 4 I showed would be part of the systems that I illustrated in East Africa, or parts of that milpa system in South in Central America. My predecessor, Dr. Adams, developed what he called a bean idiotype, which was published in 1982 here, and he shows a diagram of what he felt we needed for a new bean idiotype. And you can see it's very much an architectural improved type, an upright type with improvements in plant height, taller bean, a little longer maturity to get a larger plant, greater stem strength for lodging resistance, and a deeper taproot. So these were part and parcel of that bean idiotype. And we've been working on developing that in most of the seed types that we currently raise. And I'm illustrating that here now, these idiotypes in the Mesoamerican navy and black bean races, in the Durango races, which were traditionally vine type 3 or prostrate types. We've now developed upright Pinto and Great Northern, and likewise upright small red and pink types. These types then provide us some level of disease resistance. These upright types give us what we would call an architectural avoidance to certain diseases like white mold. And here I'm illustrating an upright type in the upper left slide versus a decumbent type, which you can see is near touching the ground and would have much more incidence of white mold. White mold is caused by sclerotinia. And the sclerotinia on the bottom here is not a bad writing of my bad writing with a magic marker. These are written with the actual sclerotial bodies that you can collect of white mold from an infected field. The other advantage, excuse me, of the upright types is that we've been able to change our bean harvest from a three-step process where the conventional bean harvest involved pulling or knifing the beans when they were slightly immature and allowing them to dry and then coming back later to pick them up and harvest them. This involved a number of obviously more equipment, more labor and so forth, but this was the only way to effectively harvest certain decumbent plant types. So from that, with these new upright types now, we have been able to go towards something of direct harvest where we refer to maybe as clipping or straight cutting. Again, we have savings in labor, equipment, and time as a result of that. We get improved quality because we're not bringing soil up into the product, and it gives our farmers more flexibility in terms of what they can harvest, and it allows them to plant and handle more acres in a day. I mentioned canning quality. All beans have to be processed in some way before they can be consumed. So canning quality is very important. And here you can see a series of different seed types that have been blanched and put in the can. Now we're in the process of adding brine to put these in the retort for canning. You can see a range of types from white beans, black beans, through pintos and red beans. As we look at selection for canning quality, our selection is done almost exclusively on visual selection. Since we're handling hundreds of lines, we don't attempt to taste these. Most beans are sold in other products with different flavoring, so we're looking, as the consumer eats with their eye, we're using trained judges to visually select. And I've listed here the traits that we're looking at: whole seed integrity, color retention, uniformity of color, clear brine, and so forth. And as we advance materials, then we do more in-detail measurements of color with a Hunter color meter. We measure hydration and drain weight ratios. These are important characteristics for the processing industry, and texture with the Kramer shear press. So this is a series of the traits that we measure for quality. And the next slide illustrates an example of a bean on the left that shows good seed integrity versus one on the right where you see a lot of splitting and breakage. This is not because the bean was handled, these were handled the same prior to harvest. It's just that certain varieties hold together better and show much better seed integrity than the others. And one that breaks would be obviously eliminated from our program. Colors, again, critical. Color in black beans is very critical. Here we're showing some canned black beans, 3 varieties, and we're measuring color with this Hunter color meter. So the lower the number, the darker, the blacker the bean, and that's what the processing industry wants. And you can see a 1-point difference on the lightness scale can be detected by the human eye. And you can see the bean in the center, which is a new variety, has a lower score, hence a blacker bean, something that the industry wants. They don't want something that has washed out color when the consumer opens the can. Disease resistance, beans are attacked by a range of different pathogens. Those that have the light green color, like anthracnose, common bacterial blight, and bean common mosaic virus are all seed-borne diseases and are particularly problematic for us, not only us in Michigan and the U.S., but also for subsistence farming systems overseas. So it's important to have disease resistance to those diseases that are spread through the seed. Diseases like rust, angular leaf spot, and white mold. Again, white mold is a very serious problem in Michigan. Rust and root rots, again, can be problematic in certain seasons. And these are some of the diseases that we work towards in terms of developing disease resistance or incorporating disease resistance into new varieties. Here I'm illustrating an example of anthracnose, where the field on the right of black beans, which is a variety that's resistant to anthracnose. You can see, if you look at the picture in the front, you can see healthy pods versus an older susceptible navy bean variety where the farmer planted infected seed. That field was completely infected, and you can see the lesions on the pods. Resistance to this disease is controlled by major genes. It's quite easy to effectively screen, as I show in the middle of the slide, in the seedling stage. But again, it's a highly variable pathogen, and we're looking at ways to improve more the durability of resistance to a disease like anthracnose. Again, an example of common bacterial blight in the center. I'm showing an example of early infection on some materials, a healthy variety with the coded number in the center. And then as you see at maturity, you see the healthy beans with the clear pods on the left-hand side of the screen, and the infected leaves and pods on the right-hand side of the screen in terms of susceptible to this particular disease. Again, these are diseases to which we have genetic resistance, and we're in the process of continuing to incorporate these into new varieties. In terms of tools to assist us in our phenotypic selection, we have gone to using marker-assisted selection. We've been involved in developing markers over the years for many of our major disease resistance genes here. And as I illustrate in this slide, here we have a series of anthracnose resistance genes to which markers have been developed both in our lab and in other labs, to the CO genes for anthracnose, the BC genes for bean common mosaic virus, or the UR genes for rust resistance. So we have markers, and the markers I'm referring to are generally scar markers at this point in time. They're very user-friendly, and they've been widely deployed by breeding programs, not only in the US and North America, but in other countries, to develop resistance to anthracnose, common blight, and angular leaf spot, as is illustrated in this particular slide. Again, marker-aided or marker-assisted breeding has been important for these diseases, and I just list them here again, more to illustrate the names of the pathogens which I hadn't included in an earlier slide. And we have, over the years, been able to develop pyramided resistance to both rust and BCMV by using combinations of marker-assisted selection and direct selection, where we've been able to combine these to get multiple levels of resistance in both pintos and Great Northerns and navy beans for resistance, as I indicate, to rust and to bean common mosaic virus. More recently, with financial assistance from the Bean Cap project, the project has developed a gene chip, which is listed here as the Bark Bean 6K chip. It's a chip of 6,000 SNP markers. This is our first access to SNP markers, and one that was very much, the Bean Cap was very influential in helping us develop. Since the community was very used to using scar markers, part of that also was at North Dakota State University, they developed then a whole series of indel markers that could be used by breeders in small programs, and there's over 2,600 indel markers now also developed for use by bean breeders. So this is some of the more recent transitions that have occurred with the assistance of bean breeding. This has allowed us to expand some of those technologies in our international programs. And here I'm just illustrating an example of this is some of our work through Feed the Future Innovation Lab, known as the Legume Innovation Lab, where we're working to improve disease resistance and productivity in some of the Andean varieties. And we've been able to move some of those marker technologies to local programs and allow them to develop, again, more disease-resistant materials for their local production area. And then similarly in projects with the same funding in Rwanda, again, where beans are very much a crop that's grown by the women of the women heads of the households, and also marketing it in the marketplace. Again, developing more disease resistance in the climbing beans, which are particularly well adapted there, has been a part of our effort too in these international projects. I'm going to finish there with a reminder that beans are a nutrient-dense food. They're good to eat, or regardless of whether you call them beans or frijoles or whatever. They're good to eat, they're good to enjoy, and I certainly encourage you to consume more beans. Before I finish, I'd just like to illustrate then some of the funding sources. First of all, I want to recognize the MSU Bean Breeding and Genetics team, my colleagues here, all the graduate students that worked in the project, my predecessors who have contributed to bean breeding not only at Michigan State and beyond. Our funding sources through Michigan State University, and also international funding through USAID for the Legume Innovation Lab and the Bean Cap, which has helped move beans into the genomic era. And for those of you interested in some additional reading materials, I have illustrated some websites here which you could access. The Bean Improvement Cooperative is a voluntary organization run by those interested in bean improvement, breeders, geneticists, pathologists, whoever, where we provide quite a bit of information on the genetic aspects of beans and so forth. And then some of the Bean Cap videos that have been developed on plant architecture and plant breeding science, creative and problem-solving videos on the importance of plant breeding that has been developed as part of Bean Cap. And then finally, I just want to point out that the bean genome paper was released about a week ago in Nature Genetics, and it's finally indicating and providing information on those of you interested in more of the work, the sequencing of the bean genome, and some of the diversity and some of the gene pool diversity and the domestication interests and aspects of beans. So with that, I will end and turn the microphone back to Shelby.